Welcome everyone. Just give a few seconds for the rest of the class to join before we start. All right. Looks like there aren't so many students today. Well, maybe they will join us later. But I do have a few announcements for today before starting the class. First is that I will be holding office hours right after the class at the end of lecture. I will be using the other Zoom link, the one that's not recorded, and that's for my office hours. That's also available on Canvas. So if you have, there's anything that you would like to discuss, come speak with me after class. The other thing I wanted to talk about was, just to clarify the final project, I have cleared the website and put the deadlines and everything should be in place there for you to see. So if you haven't looked at that, definitely encourage you to do that. So there is a form for you to fill if you have not, maybe you have already, but I'm gonna paste it again in the chat for everyone to look at it again in case you haven't. I am requiring that one person from each group fills the form. So if someone from your group has already filled it, then you don't need to fill it again. But if you're working in alone, or if no one has filled it yet, then you should fill it as soon as possible, because that will help us decide how to schedule a lecture for next Friday based on the presentations, and also so we can keep track of everyone's teams and projects. So I will give you maybe 30 seconds or 1 minute to fill that form right now. I see there's a few more students joining us, so I will paste the form again, and I will give you maybe 1 minute, and then we'll start lecture. If you've already filled it, then of course you don't need to fill it again. You just have some time to relax now. I might, because some new students are joining, I'm going to share my screen as well. All right, with that being done, we can actually start our lecture. So today's lecture will be pretty short because, as you already know now, we have a guest lecture starting in just about 25 minutes or 20 minutes at 3:30. So I will tell you a bit about that. You should right now just be able to see my slide, and this is our last official class together because Wednesday is the R lab, and Friday I won't actually be lecturing because it will be the project showtime. And for this last session, in the half hour or so that we have, I'd like to talk about a fairly important, very relevant for modern days topic. And this is something you may think that you might have never heard about. You might not find this in a statistics class, but I will try to convince you that it is important. And as it turns out, most people think that science is a reliable way to answer questions about the world. For example, when a doctor prescribes a treatment, and that treatment we trust because that it has been shown to be effective through research. And we have similar faith that the airplanes that we fly in aren't going to fall from the sky, and so on. We have really this confidence that science and scientific findings drive technological progress and well-being for all of society and mankind. But there's been in the last decade or so, there's been some increasing concerns that science may not always work as well as we have long thought that it does. And in this section, I will discuss some of the concerns about the reproducibility of scientific research and outline some of the steps that you can take to make sure that your statistical results are as reproducible as possible. And this is perhaps an outline of how we think that classical science should work. So let's say we're interested in a research project on how children choose what to eat. And this is a question that was asked in a study by a lot of researchers. I'm giving an example here where the question starts with this hypothesis. The hypothesis here is that branding with popular characters should cause children to choose healthy food more often. Then you collect some data and you do an experiment. You basically run your own simulation to get in real life by offering children the choice between a cookie and say an apple with either a control sticker or some fancy brand sticker. And then you record what they choose. And then you do statistics to test the null hypothesis. And the null hypothesis I stated here, it's pretty long, but it is, it has a few, it's pretty, it involves numbers and everything. Why does that, and this is, and this is the conclusion that you might find. The pre-planned comparison shows this results and this association with an increase. When you say the p-value, you conducted some chi-square tests. This is the results of your chi-squared. And then you make some conclusion based on that data. Maybe the conclusion could be that the study suggests that the use of branding may benefit healthier foods more than they benefit the indulgent or highly processed foods, and so on and so forth. And the researcher who actually conducted this study, Brian Weinsick, he's well known for his books on mindless eating, and he also has a pretty large fee for corporate speaking engagements. And in 2017, a set of researchers began to scrutinize some of his public research. They started with a set of papers about how much pizza people ate at a buffet. And the researchers asked Weinsick to share the data from the studies, but he refused. So they had to dig into his published papers, and they found some number of inconsistencies and statistical problems in the papers. And this publicity led a number of other researchers to dig into this work and so on. And I won't go into all of the details here, but it turns out that at the end, in 2018, 15 of the research studies were actually retracted. They were like published papers that were kind of canceled, and he actually resigned from his faculty position at Cornell University. So I'm showing here an example where in that paragraph here where I have this yellow highlighting, if you're doing some analysis but you're not getting the results that you were hoping for, say ideally you would like the results to be significant, because that's the entire point of your paper. If you don't have a significant result, you have no paper to write. Well, then you really wanna think harder, and you really wanna convince yourself that it should be, this p-value should be lower. So you might ask your students or collaborators, do you want to take a look at it and see what you think? Maybe if you can get the data and maybe if it needs some tweaking, then it would be really good to get that p-value below 0.05. And you might think that this kind of behavior is pretty rare, but actually it's becoming increasingly clear that problems with reproducibility are very widespread in science. There's a large number of papers that have been written in the last 5 years where researchers took hundreds of published studies in psychology and attempted to reproduce the results that were originally reported in the papers. And their findings were shocking, that in 97% of the papers, the claim was that reporting some statistically significant findings, but only 37% of these effects were successfully replicated and significant in the second replication study. And I'm taking here the example of psychology, but really this sort of issue is present in almost every area of science, from cancer biology, chemistry, economics, social sciences, and so on. So this is a very, very serious issue when so much effort and time is devoted to this research, and that's clearly problematic when after that you might go and try to actually, what you care about is the truth, and you don't just care about having a fancy paper at the end of the day. So there's a lot I could say here about this topic, and what are some of the biases that lead to the issue that we see here, and that there isn't just a clear one single reason behind this reproducibility crisis, but one reason for it is actually a cognitive kind of bias that sometimes, when you've invested so much of your time and efforts and energy into a project, then you really want to convince yourself that it's actually significant, and then you will pay more attention to information that confirms your hypothesis than to information that disconfirms it, although both are publicly available and they're out there in the wild for you to read. Kind of become selective about what you agree to read and what you decide to rather overlook. So there's a lot of recent work on this and how attempts to really understand and fix this issue. Most recently, one thing you could think about is this all just due to the statistical framework? Is it a problem with statistics that we're doing null hypothesis statistical testing, and in this framework we've seen some of the limitations of it, and is it actually the cause behind this epidemic of false results? In fact, in just, I think this was in 2005, there was this paper by John Ioannidis, physician from Stanford, arguing that the use of null hypothesis statistical testing in the context of modern science would necessarily lead to high level of false results. And his analysis included and focused on the concept of positive predictive value, which is defined as the proportion of positive results that are true. Positive results means here that your test found them to be significant. So if you remember a couple of weeks ago, we studied type 1 error and type 2 error. And this is about type 1 error, when you're under the null and you're looking at positive results. And the complement of that is actually true positive results. So it's 1 minus the type 1 error. And it turns out you can do a certain calculation here that actually, if you think about it, only involves Bayes' rule. And assuming that we know the probability that our hypothesis is true, and you can do a two-line calculation where essentially your results depend on some number of baselines. You basically need a baseline about, say, the probability of your hypothesis being true. And that's something you'll often see whenever you do Bayesian testing. You need some kind of prior. So in this first example, let's assume that you're in a certain field, say, where the hypotheses being tested are true 80% of the time. This is just say a guess that we're making. Then based on this, and well, you can actually go and compute this proportion of positive values. So what this means here is that if we find a positive result in a study where the hypothesis is likely to be true, and when power is high, then the likelihood of it effectively being true is pretty high. But if you go to a different maybe field where the now the probability, the prior probability of the hypothesis being true before even looking at your data is pretty low, and that can vary from field to field. Then in this case, the PPV, the probability of the positive predicted value is substantially lower. So this means that in a field where most of the hypotheses are likely to be wrong, and of course that's an interesting scientific field where researchers are taking risks and testing hypotheses, although they might not be true. Then even when you find a positive result, well, it's more likely to be false than to be true, because this positive predicted value is less than 0.5. So this is just another example of how important the base rate effect is. And we discussed this in the context of hypothesis testing. When an outcome is unlikely, then it's almost certain that even if you have positive outcomes, then most likely these will be false positives. And you can do a number of simulations on this. I put a notebook on the website if you're interested in digging into these kind of scenarios better, although they're all based on simulation. And the truth is that I mean, statistical power, which is the ability to really find signal when there is signal, is pretty low in a lot of settings. A lot of hypotheses that are being tested, but findings that can actually replicate are not so easy to come by. So unfortunately, we are in the setting of the second scenario where the PPV is pretty low. And I mean, you know, this is the sort of thing that's quite systematic. That I mean, generally, when you read a paper, you have to take it with a grain of salt, and you should assume that the results that are stated in the paper are somehow inflated. That is unavoidable. This is another simulation where we look at the true effect size and the mean effect size of significant effects, but I just, what you basically see here is that the true effect size is, I mean, systematically lower than the one of the significant effect sizes, and there's a precise statistical reason behind it. And now I would like to move to some questionable research practices. There's this popular book that was published in 2004 that aims to provide aspiring researchers with guidance on how to build their career, and in one of the chapters there, that's titled Writing the Empirical Journal article, one of the authors provides some suggestions about how to write a research paper. And unfortunately, some of the practices there are fairly problematic. You can even give them a name. You can call them questionable research practices, QRPs. In this first example, I'm showing which article should you write when what the author suggests here is basically known as hearken. That comes from hypothesizing after the results are known. And this might seem innocuous, but you only claim and you only look at your hypothesis after looking at your data. And that is actually a dangerous practice because you're reframing your research and your conclusion as an a priori prediction. You're claiming that the question and the hypothesis you made before even looking at the data, and magic, it turned out to be true. Whereas what happened was actually the opposite. So this essentially allows the researcher to rewrite their theory based on the facts, rather than using the theory to make predictions and then to test them. So it becomes very difficult to disconfirm incorrect ideas, because the goal can always be moved to match the data. And in the second paragraph, what the author suggests is known as p-hacking, which basically refers to trying many, many different analyses until one finds a significant result. And of course, it is correct to do it if you report every analysis that you do on the data. But it's very rare to find that, actually. In most papers, you only find an analysis or two that will be reported, which are the ones that came as significant. They only present the analysis that worked, which usually means that they found a statistically significant result. And there is many ways to continue on this trend, and I put here some of the illustration of the dangerous consequences of and nefarious consequences of dealing with this. You can even do a formal mathematical analysis of how much you're losing when you're doing this kind of following this approach. But the consequence is basically that these p-hacking strategies greatly increase the actual false positive rate, and they result in a higher number of false positive results. So this is a funny exercise that I encourage you to do. You know, you just go to this website. The slide is on the course website, and the link is there. And basically, if you could think, if your name ends with an A to L, I just pick those letters at random, then your goal is to try and find evidence based on the data there that the US economy is better when Republicans are in office. And if your last name starts with M to Z, then find evidence for the opposite conclusion. And we don't have the time to do it in class right now, but I definitely would love to see the results of this analysis. It's basically you can make the data say whatever you'd like it to say by looking at a different variable, by defining the data slightly differently, by removing some of the data, and so on. And of course, I'm not taking position here about which of the two claims are true and which is wrong. This is beyond the scope of a statistics class, but this is a pretty good example where you can find evidence for one topic and then the exact opposite. Okay, so I think that's all I had to cover for today, and there's actually a whole lot more to say about this. I was pretty brief in the way I covered everything. This is just a few slides. So I actually posted a more complete set of slides on the course website that goes over a lot of other more examples that we won't necessarily have the time to cover today, but that could be enlightening for you to see. And this is not something you will be evaluated on, of course, but it's something that that goes way beyond the scope of a single class. This is I purposefully decided to put this towards the end of class rather than towards the beginning. Although the concepts here are fairly simple, you only need to know what a p-value is and what hypothesis testing is. But it really is a key thing to be aware of as you go in the future and use these skills and maybe go and continue to do data analysis in the future. This is something to really be aware of, that you might be in settings where you're not actually trying to replicate other researchers' findings, but you just want to make sure that the research you're doing is actually valid, reproducible research. If someone goes on and tries to reproduce it, they should be able to. And that, of course, has several other, involves a lot of other things. You also usually want to make both your data and your analysis code available on the internet. That just makes it easy for everyone to actually go and confirm for their own self. And other researchers can try to reproduce the results and potentially test different analysis methods on the same data. So this is a very important move that's quite recent towards just making sure that there's open sharing of code and data. And it takes a lot more than just a lecture like this to become aware of this. There's a lot of incentive structures as well that might need to change to make sure that researchers are actually have good incentives to provide this sort of completely shareable research material, and sometimes might ask them to pre-register their studies. But it's definitely something we strongly advocate for in general. And there's many ways to share your data and your code. We ask you to share your data and your code in this class as well. We try to make everything open and give you access to the code we use to generate our markdown files and so on. Okay, so that's all I had to say for today about how you might take a step towards doing better science. And it really is every scientist's responsibility to improve their research practices in order to increase the reproducibility of their research. So it's not just about finding a significant result, but it's really about asking and answering questions about nature in the most truthful way possible. So most of your hypotheses may be wrong. That's the nature of your research, but you should be comfortable with that. So, and then when you find one that is right, you will actually be even more confident in its truth. And for the rest of lecture, I would like to introduce Professor Tibshirani, who has been a strong advocate for reproducibility. And this is something he could also speak more extensively about, but I think he will talk about another topic for today, which is more of an introduction to data science. In case you've never met Professor Tibshirani, he's a professor of biomedical data science and statistics in both departments here at Stanford. He co-authored the first study that linked cell phone usage with car accidents. That was a widely cited article that has played a key role in the introduction of legislation that restricts the use of phones while driving. And he's one of the most influential statisticians and scientists of our time, so it's a true privilege to have him here in class today. Thank you, Rob, for joining us, and I will give you the floor. I believe you will be able to share your screen once I stop sharing mine.
Okay.
Because you had a few slides, I think, for us.
Okay, so hi everybody. Thanks for the kind words. I mean, I'm Ismael's PhD supervisor, so that's one of the reasons why he had to say all those nice things. So happy to be here, and I'm going to give you sort of a general talk about data science and statistics, and then tell you about a couple of examples, which a couple of collaborative projects which will illustrate some of the exciting things about statistics and data science and some of the challenges. Now, I'm really relying on you guys to ask questions, really, because a lot of this material will be more advanced in detail than you've seen in this course. So I don't want to just spend, an hour talking to myself and have it over your heads. You really have to stop me with questions. There's no, as I always tell my students, no question's a dumb question, okay? So please stop me, and I'm going to try to draw out some questions along the way so it'll be useful to everybody. So let me share the screen. And I sent the slides to Ismael. I don't know if did you send them on, Ismael, or you will?
I will make them available on the course website right after lecture.
So everybody can see this, I guess. Yeah, I'll make it. Let's see. Okay. So we can all see that? Yep.
Great.
Okay, so I'm a statistician in the Department of Statistics, where Ismael is a student also in biomedical data sciences at Stanford. So it means a lot of my work is very applied, and especially applied to biomedical area and health, which is exciting because you feel like you can potentially make a contribution to public health and medicine, which is important. And that's the core. Okay, so what am I going to talk about? Just some general thoughts about data science and health. Quite honestly, I'd like to get you excited about the field of statistics and data science, which is a very, very hot field right now. Like, this wasn't true I was a graduate student, how many years ago now? 30 years ago, more. And back then, statistics was kind of a dry science that people, if you told me you're a statistician at a party, they would run for the door. Like, they don't want to talk to you. But now you say you're a data scientist. Oh, really? Data science? That's interesting. Where do you work? What kind of problems are you working on? All of a sudden, data science is really exciting and hot to people. So that's a great thing. So it means there's lots of jobs in this field, especially in health, where we collect data a lot. In health, data is collected a lot, and there's a lot of information we get about patients at many, many levels. Clinical measurements, genetic measurements, and we can use this data to try to make better decisions about their health outcomes. We talk about something called supervised learning, which is a lot of what statisticians and data scientists do. And then I'm going to give you two examples of it in my own work. One is in a blood-related project at Stanford Hospital, and more recently, some COVID-19 forecasting, which I've been involved in. Okay, so as I mentioned, there's a lot of excitement about data science and health. And you'll hear the terms artificial intelligence, predictive analytics, predictive medicine. All these are to do with really using information to try to predict patient outcomes, to choose better treatments for patients, et cetera. And there's a lot of data available now. So what's really changed in the last 20, 30 years is the technology, right? In the old days, you collect a small number of measures from a physical chart. Doctors would. And now, of course, we have MRI machines and CAT scans. We even collect data on patients from smartphones now, right? That's quite common on people. And we can try to use the information we get on the smartphone to figure out how much are they exercising, for example. How does their exercise level correlate with their blood pressure, et cetera? So there's a huge amount of very complex data that's coming that we have to make sense of. Okay? That's a huge opportunity. And this, whenever I give a talk, a sort of a general talk now, I quote this Hal Varian, who I know is chief economist at Google, who said, in 2009, he said, the sexiest job in the next 10 years will be statisticians, right? This is wonderful for statisticians to hear this. And it's been sort of repeated by Harvard Business Review. Whether we're the sexiest people, I don't know, but at least there's a lot of interest in data science and statistics, a lot of demand for our services. Okay. So now I want to explain what supervised learning is. And I'm going to explain it with this, first of all, with this abstract picture, and then I'll show you a very simple example and come back to it. But a lot of what statistics data science is we try to predict something, like, for example, try to predict how long a patient's going to live from things we know about the patient. Okay, and that's called supervised learning. So typically we have what's called training data on the patient. So for every patient, like John Smith, we have whether or not, let's say how long they lived after a heart operation. We also know about John Smith. We know features about John Smith: his age, some clinical measurements, blood pressure, pulse, etc. Maybe some genetic information about John Smith, whether he has certain genotypes which make him more or less likely to have heart disease, etc. And supervised learning basically tries to predict the target or the output, like how long the person's going to live, based as a function of the inputs, right? Prediction is sort of a basic thing that's done in science, and it's a major challenge for statisticians and data scientists. Predict the quantity of interest from things you know about the patient. Okay, so supervised learning, we have a bunch of training data. We have, for example, we have patients, John Smith, Jane Doe. For each of the patients, we have the actual output, say how long they lived. We also have things we measure about the patient, and we try to build an equation, basically, that predicts the output from the input. So that if a new patient comes to the hospital and the doctor asks us, how long is this patient going to live after the heart attack, we can put his or her measurements into our equation, and it'll say, doctor, this patient's only going to live for 3 days. And the doctor might decide to use, maybe to give this patient more drugs or more aggressive treatment. Okay, so this is the basic idea of supervised learning. You have training data, you have what's called a learning algorithm, which is some way of forming an equation that predicts the output from the inputs, and then we want to use it for new data, for a new patient, or whatever our domain of application is. So let me, first of all, I want to stop right there to see if there are questions, because if I lose you at this point, then the rest of the talk will be useless. So please ask questions. Is the sort of the basic paradigm? I haven't told you how we do this, but I just want you to understand what the goal is, what the construction is. Don't be shy. If you have a question.
How certain is it that the actual result is correct?
So the actual result in the training data?
Yeah.
Oh, well, that's a good question. Sometimes, I mean, we assume basically that there's no error in say this is how many days the patient lives. We assume that that's been recorded correctly. There are situations where you worry about the data actually being recorded wrong, but that's sort of a more complex problem. Here, we're at least assuming that the data we have is correct, and our challenge is to try to figure out a good prediction equation to predict the output from the input.
And if a wrong data, like let's say you didn't get the correct amount of days, how much will that affect the algorithm?
It's a good question. Some algorithms are quite robust in that sense. There won't be much affected. Other ones will be badly affected. That's actually a very good question, because one of the examples I give later with trying to predict how much blood is going to be needed by Stanford Hospital, I think bad data actually is causing us great problems. So what you're saying can be a real problem, not only the outputs or the inputs, right? If this is from a patient chart and someone transcribes the wrong number, their blood pressure was actually 120 and was marked as 20, right? The algorithm could be completely messed up by that. That's a very good question. And depending on the algorithm, that can be a real problem. Yeah.
Okay, thank you.
You're welcome. So again, I guess, is this a new thing? Is this like something we just invented this year? No, this is not at all new. This sort of basic way of reasoning, of building prediction equations from data and using them on new data, that paradigm's been around for a long time. But why all the excitement now? Well, it's because of this. In the old days, people would build these training sets sort of from scratch. So suppose you're trying to predict, again, the chance of getting heart disease from things you know about the patient. In past years, you talk to a cardiologist and they say, well, what's important clearly is their family history, heart disease, their age, their cholesterol level. They might find that through a lot of different studies, they pick the top 10 things they know about, they think are important. And the inputs would just be 10 things we measure about patients. So that was sort of the old way of doing things, sort of pre-computer age, pre-technology. And even a lot of these rules sort of exist today, these scoring systems, but they're kind of antiquated. What's changed now is that we measure many, many things on patients through like scans, PET scans, MRI scans. We get their all their gene measurements, for example, all their mutations, right? And now the number of inputs has gone up from 10 to maybe 10,000. So and now the algorithm has to, first of all, it's an opportunity because there are many more things from which to build the equation. We have to sort our way through, most of these inputs are probably not going to be useful, right? You're talking about the genome. If I know sort of every mutation that's in your genome, the great majority of them are not going to have any effect on your chance of getting heart disease, right? There's probably a few genes in certain places which, if you have this mutation, you're more likely to get heart disease. But figuring out where those are is a real challenge. So what's changed is the whole scale of this. This is much bigger. The data is much bigger than it used to be. We have many, many more features we measure about patients or whatever our unit is. And the algorithms are much more sophisticated because they need to sort their way through all the inputs, find the ones that are predictive, and throw the ones out that are not predictive. So it's called supervised learning because, well, learning, it's called learning because the algorithm is learning how to predict the output from the inputs. Right? It's almost like a human brain. It's trying to figure out what's the best prediction equation to predict output from input. It's supervised because it has examples of the true output. So again, in the patient example, every patient we know not only the features we've measured, but we know how long they actually lived after their heart attack. So we're giving examples to the algorithm. It's supervised, and it uses the examples and the inputs to figure out a good prediction equation. Okay, so this is sort of a high-level thing. But what does this come down to? Very, very simple. Here's the simplest supervised learning you can imagine. Okay, this is a single feature now. So what we have here is actually took this picture off the web. We have for 15 students, we have their GPA score, and we have their ACT score. Okay, that's each of the black points. Okay, and our prediction job is to say, our task is to predict, given the GPA, what's the student's score on ACT going to be? Okay, that's the job. And this is our training data. It's just these 15 scatter points here. Okay? So how would you make such a prediction? What prediction equation would you use? Well, the simplest one is use a straight line fit. So I've put this straight line through the data points. This is called the least squares fit. Am I right that you guys have not seen this in the course before? Yes, no, somebody?
We haven't seen this graph before.
Okay, well, good. This is really important, and it's simple. It's called the line of best fit. So this red line has the property that if I take the sum of squared deviations, each point to its prediction on the line, from here to here, here to here, add up all those squared errors, it's the one that has the smallest it's the best approximator. It's the line of best fit. In some sense, it goes right through the data points, right? Clearly, this has a better fit than if I had the line sitting up here, right? So this red line is a line of best fit. It's called the least squares line. It's the simplest example of supervised learning. I take the training data, I construct the red line, and now if you give me a patient, you say, Rob, student, I've got a student with a GPA of 3.6. What do you predict his ACT would be? Well, I'll go to the line, I'll say, okay, well, 3.6, that goes to about 31. That's my prediction. That's supervised learning in its simplest form. I want to stop for questions right there, because is that clear? Well, here we have one feature now. Simplest thing, one feature, GPA. We're trying to pick the target ACT. We have training data, the black points. And our prediction equation is just this line of best fit, the least squares line. Please tell me if it's not clear. Don't be shy. Okay. So this is a form of supervised learning using one feature. Very simple. Okay. So now we're, of course, using one feature is not that interesting, right? Because I mean, if we're trying to predict how long a patient lives, we're not going to use just one thing we know about the patient. Their age, for example, is not enough to, not enough information, right? Their age is probably, the older they are, probably not going to live as long, but their blood pressure is probably important for heart disease, cholesterol measurements, right? So clearly we want to use multiple features. We have to walk before we can run. So that's why I started with just the one feature example. But now, in general, we're going to have a prediction equation involving p different features. So now, here's a little notation. Here's our training data, again, just as before. But now each of these x's is not one number, but a whole set of numbers, a collection of p features. These are all the things we measure on a given patient, for example. Maybe there's 10 such features. Maybe there's 1,000 such features, whatever. But this is for the first patient. Here's his or her measurements. Here's his or her outcome. And for patient number n, all his measurements, his outcome. That's our training data. So now before we fit a line through the data, now we're going to fit what's called a hyperplane, or basically we're going to fit a prediction equation which has got a slope for each of the features plus the intercept. Here's my prediction equation. And the x's are the observed features, the y is the outcome. And now here's this least squares criterion. The line of best fit is now replaced by this. For each individual, here's the actual outcome, like a lifetime. Here's the prediction, right? Now we're going to choose the weights to give the best fit. In the same way that here we chose the slope and intercept of the red line to produce the best fit, right? That's this line of best fit. Same thing here, except we have more than one feature. We're just going to choose the weights to give you the best fit. Now is to make the discrepancy between what we're trying to predict and our prediction as small as possible. Okay, so now I'm really going to stop for questions. I'll bet there is questions.
If multiple factors is like affecting the result, how can we, when the computer is computing or when we are researching, how can we make sure that, how can we make sure that this is the effect of one factor but not the other factor?
Didn't quite get your question. So we're talking about this procedure here where how can I be sure that
like in total,
yeah,
focusing on this equation,
mm-hmm.
Because in real world, multiple factors is affecting on the result, and when we are researching, how can we make sure that the result is effect is based on one of the factor but not the other?
Well, I think I understand your question. I mean, we have our training data. Now, let me ask you, suppose this, so we're going to choose the weights to make this fit as good as possible. Suppose this feature was, again, suppose we're predicting how long they live after a heart attack. Suppose this first feature is like the family history of heart disease. Okay, and this second feature is, I'm just making this up, their shoe size, okay, which we measured for some reason, I don't know. So if that was the case, would you expect, and we're going to find the best weights, would you expect this weight, the weight on family history, or the weight on shoe size to be more important? So to be larger, right? We're going to choose the best weights. Some of the weights will be close to 0. Some of the weights would be chosen to be quite large, right? We're going to choose the weights for a feature that's quite predictive, like family history. We're going to choose a large weight, right? Because it's going to be important in the equation. For a feature which is, maybe I should have said instead of shoe size, which could actually be relevant, hair color. Suppose this was hair color, red, brown, black, red. You wouldn't expect hair color to be predictive of how long you're going to live, right? So the weight on hair color is probably going to be close to 0, right? So I don't know if I'm answering your question. By choosing the weights to produce the best fit, it's automatically going to choose large weights for things which are predictive and small weights for things which are not predictive. Is that clear or?
That's clear. Thank you.
But I mean, a high level, was there a question or?
Yeah, I have a question. So I'm seeing training data. So that's assuming that we have training data.
Yes.
What do you do in instances where like the data just doesn't exist? So I'm like, sounds like you're waiting years to collect all that information in order to come up with this predictive model in the first place. Or are you looking at past like, yeah, I'm just wondering like how many years people are waiting to actually get all of this data to actually create this model in the first place.
Well, that's a very good question. So you need, to build a predictive equation, you need all this data, right? And sometimes, right, sometimes if it's what's called a prospective study, you'll actually have to wait, you know, maybe you're running a clinical trial that you don't hear the outcome till 5 years. You'd have to wait for the 5 years to get the data. More often you use historical data. Right. So you use data which has been collected in the past on patients, so, or whatever your domain is. But so that has a big advantage that you can use it right away. So now, like if you look on the web, there's many websites that have collections of data of this form in all kinds of domains, in medicine, finance. Here's another example. You're trying to predict stock returns. Why? From things you know about a company, right? It's how much profit the company made, what its market share is, how many employees it has, right? So a huge lot of work is done in the financial industry to try to predict stock returns for a given stock based on things you know about the companies. So that on the web, there's huge databases of training data for companies, right? And for this data, it's all been pre-compiled for you, and you can, for this purpose. So it's a good question. You need training data. A lot of it's available historically.
Thank you.
Okay. So if you don't understand the details of this, the more important thing is you sort of understand sort of at a high level that we're going to take data which is with features that we know about patients or companies, and we have measurements on something we're trying to predict, like the stock returns of the company, or for patient, how long they will live after a heart attack. And we're going to build a prediction equation and apply that to future data. That's sort of the high level, actually. And a lot of what statistics data science does, a lot of it falls into this framework. Another example is self-driving cars, right? Self-driving cars use supervised learning all the time because it has cars to figure out, you know, in a given situation, if I turn the steering wheel by a few degrees, what's going to be the result of what's the car going to do, right? Under various conditions. So again, you have labeled data, and the cars have been trained with labeled data, so that in the future, when the car has to make a decision, you know, I want to turn right here, and the road's got a certain slope, and the model will be able to. The model will be used. The prediction equation will be used to guide the car. So this is a very, very useful, very common task that's done in data science all the time, in machine learning. Okay. So here's a question for you. I say least squares doesn't work if the number of features is greater than the number of observations. So what happens, I've got n observations here, right? Like n people, and each of these has got p features. Like here I had 15 people, right, and 1 feature. What happens if I had 15 people and I measured, each one I measured like 20 things? What would happen to this least squares? What would be the result of, is this least squares going to, is there a problem with least squares? I claim there's a problem with doing least squares. Do you remember back from your algebra, maybe, if you have a set of equations and you have more unknowns than you have equations, what happens? Like you want to solve a system of equations and there's like 5 unknowns and there's only 3 equations. Anybody remember what happens? No?
There will be leftover unknowns.
I'm sorry, well, there's leftover unknowns, right? What happens is there's no solution, there's no unique solution to the problem. It's overspecified. in this scenario, what it means is that actually there's no unique set of weights that minimizes this, and basically the system, it overfits. So you have, it's going to produce zero error, but will not be a good predictor because it's got too many parameters and not enough observations. So actually, let's not worry about that. That was a point that's not really germane to the topic. I only want you to come from this first. I want you to come away with this basic paradigm, and I'm going to show you two examples of where I use this paradigm in real collaborations. This supervised learning. Okay, so two examples. Oh, I mentioned deep learning. You might have heard of deep learning. It's another fancy way of doing supervised learning. So just for your reference, there's some details about that here. We're sure not going to talk about it, but deep learning is a very hot topic now in data science. So now let's get to two real examples of supervised learning with real data for medical purposes. Okay, so platelets are a clotting factor. They're a component of blood, and they're responsible for, they clot your blood. Like when you cut yourself, your blood clots to prevent further bleeding, and platelets are what cause you to clot, which is good. This is actually a unit of platelets. It's actually this sort of yellow color. Very valuable part of the blood. So there's red blood cells, white blood cells, and platelets are three major parts. And there's a very valuable resource for the hospital. For example, if someone has surgery, they're given platelets to prevent bleeding from the surgery, right? So a very important part of blood. So what is this project about? This is the team of people that work with me on this. Ta is the head of the Stanford Blood Center. These are medical residents, pathology, and these are two grad students who work with me and my colleague, all from statistics. So this just makes the point that a lot of this kind of team science requires a real team of people. So what is the problem? By the way, here's the paper, first paper, if you want to read about it more formally. Here basically is the problem. So I actually got contacted by Todd, the head of Stanford Blood Center. He said, okay, Stanford Blood Center supplies all the blood to Stanford Hospital and Stanford Children's Hospital, right? It's kind of unique. In most major cities, there's multiple blood centers and multiple hospitals. Stanford, we have one major adult hospital and one blood center. So what happens is Stanford Blood Center supplies the platelets to Stanford Hospital, about 45 units a day. Each of those bags I had in the previous picture, that's a unit, okay? And the problem is that platelets have just 5 days of shelf life. Okay, unlike other blood products, you can't refrigerate them. And they're safety tested for the first 2 days for HIV and other viruses. And there's only 3 days of use. So when Stanford Hospital receives a unit of platelets from Stanford's Blood Center, they have 3 days to use it. And then if they don't use it, they have to throw it out because it's no longer viable. So what happens now is it's done manually. Stanford Hospital will call up the blood center and say, I'm looking at the inventory here. I'm looking at what's on top of the hospital. We're going to need 40 bags tomorrow. Or we're going to need, 30 or 50, depending on sort of their guess as to what's going to happen in the hospital, their surgeries and things, and how many bags are currently on the shelf. But right now it's a guess. It's educated guess. And they waste about 8% of the bags, 1,400 bags a year are thrown away, which is really bad. I mean, not only is it expensive, but the people who get platelets, and I've done this myself, it's not like getting regular blood. When you get platelets, it takes about an hour and a half sitting in a chair with an IV in both arms. They remove the platelets from your blood from one arm, filter out the platelets and put them back in the other arm. It takes about an hour and a half, and you can only do it about once a month or once every 2 months because it's not safe for you to lose all those platelets. So people who donate platelets are very devoted donors, and to throw away almost 10% of their blood is really problematic, right? I mean, it should be avoided. And this problem occurs not only at Stanford, but everywhere in the world, that everyone's taking platelets and throwing a lot of them away because they've expired. So the head of the Stanford Blood Center asked me, he said, you know, you statisticians, can you help us come up with a plan, a system that to help the hospital figure out how many to order every day so that we don't waste as much. Of course, they also don't want to run short. That's even worse, right? If they run short, like if they need, all of a sudden they need 45 bags one day and they only have 35 on the shelf, they're gonna have to go to some other, there's actually almost a market for blood. They have to go somewhere else to order the blood at great cost. And sometimes they can't even find it. So they don't want to run short, but they don't want to waste it. So he asked me, you know, can you guys come up with a system to improve this? So we don't waste so much and still don't run short. So let me pause for questions because I want you to understand what the statistical challenges. I should say, he is going to give, he gives information about the hospital for like 3 years of data about the hospital. Like every day, how many patients were in each of the wards of the hospital, how many surgeries were planned, and then how many bags were used the next day. So we have 3 years of historical data of great detail. And we're going to use that to build a prediction equation to predict how much blood is going to be used tomorrow, given what's in the hospital today. That's the supervised learning task. So just so you see that this is what the train data looks like. Here's the daily consumption of units. You average around 30 or so, but it varies a lot. And then if you sum it over 3 days, it averages around 90 or 100 bags over 3 days. But there's a lot of daily variation. So does everybody understand what the problem is? You have this historical data. We want to use this historical data to train a model so that Stanford Hospital and Blood Center can use this model so that today, Monday, they can run the model and say, well, they're going to need 43 bags tomorrow. And then they deliver 43 bags rather than 50 bags and waste maybe 7. And then they don't deliver 30 bags and run out. So we want to build a system to figure out how many bags of blood will be needed each day, given what we know about the hospital. All the patients in the hospital, etc. Come on, guys, how would you do it? So before I tell you what we did, what would you do? So I'm giving you all this daily information about the hospital, all the things you can want. Every patient is in the hospital, their charts, all the surgeries that are planned for every day for the last 3 years, and then every day for the last 3 years, we also how many units of platelets were used. So what would you do?
Do a lot of t-tests to find out which characters might have like great significance contribute to the result, and then to like find equations like how exactly that result is.
Yeah, that's a good idea. So t-tests would give you some idea of whether, well, it would give you some idea of whether the individual features are important for the outcome. But what we're going to do actually more directly is we're going to go back to this prediction equation. We're going to build a prediction equation just like we said here, right? This prediction equation, whoops. We're going to fit a model like this. Y is going to be the number of platelets that are used, the number of bags of platelets used on July 1st. The X's are going to be all the things, all the features we know about the hospital and the patients. Like this first feature might be the number of surgeries that are planned. That's clearly going to be important, right, for predicting the number of bags of platelets, right? If there's like many surgeries planned, they're probably going to need lots of blood. If there's many patients, say in hematology ward, which is like blood cancers, you're probably going to need lots of blood. How about the day of the week? If this guy was the day of the week, Monday, Tuesday, Wednesday, Thursday, Friday, Saturday, Sunday, which days of the week you think would be most predictive, would correlate more with higher platelet usage? What days of the week are going to need more blood? Weekdays or weekends? What do you think?
Weekdays?
Weekdays, sure. Yeah, right. Because on weekends, there's just not as much stuff scheduled, right?
I mean, there could be people, there's more people who party, go out, more car accidents, maybe Friday night.
Could be, but it tends to be that there's a lot less surgeries planned on weekends because doctors have to have, time off too. So all else equal, probably weekdays are going to be you need more blood than weekends, right? So we're going to build a prediction equation to bring the number of the platelets we're going to use, the hospital is going to need on a given day, given lots of information we have about the hospital and the patients in the hospital on the day. So like sitting today on Monday, we're going to say, okay, we're going to have a prediction equation which will say, okay, number of surgeries planned, it's a Monday, number of patients in hematology ward, et cetera, et cetera. I may have 30 of these for each. I'm going to use this prediction equation then to say, okay, my prediction for the number of platelets that are going to be needed is 41. Some other day it might be 22, et cetera. So that's the idea. We're going to build a prediction equation. And I've got this, low-tech drawing of, let me just cut there. This is what it's going to look like. So I'm sitting today. I've got features, I call them ZI here, but I had the X before, but they're basically all the features from which I'm going to make the prediction. We have training data, 2 or 3 years of this data. We're going to build a prediction equation, and we're going to use it to predict the number of platelets needed for the next 3 days. And then we're going to combine this with knowledge of how many platelets are currently on the shelf, right? Remember, we can keep them around for 2 or 3 days to recommend to the blood center, you should supply, again, 38 bags tomorrow or 52 bags or whatever. Okay? So it's basically supervised learning done in a sort of a rolling fashion in time. Build a prediction equation to predict how many bags of blood, of platelets are going to be needed for the next 3 days. Okay, so that's it. And I won't even go into more detail except say the result was actually quite striking. So here's what, we did this in the last year or so. And this is the result of what we did. I mentioned before that that the current human system wasted about 1,400 bags a year. We're able to reduce that to, well, by 3/4 to just 339 bags per year. So the waste has gone up from 8% to below 2% with no shortage. So if you look at, this is sort of a time series of what's happening. This is the consumption of blood in red. The waste is down here, and the remaining, to turn that off. The number of bags that were left is in green. And see, the green never hits go below 0. So we're never short, but we waste far less. So the algorithm has done much better than humans. It's saved a huge amount of blood, and without having any shortage. So this is, and you might ask, well, which features turned out to be important? Well, actually, the day of the week was a big feature. Like, again, Monday to Friday is much more likely to be days when you use a lot of blood than the weekends. And some of the other things, like how many patients were in certain key wards in the hospital that use a lot of blood, how many surgeries are planned, the various things we knew about the patients. Are the patients in the hospital that have low platelets already? We're going to need more platelets. So it all made a lot of sense, and it was very effective in making the prediction. And one of the pathology residents computed that if implemented at Stanford, it could be a huge money saver for Stanford and also nationally. So what we're doing now is we've written this up into an R package. You guys have used R, right? Yes?
Yes.
Yes. Well, R is a very useful language, even for more sophisticated learning like this. So we have this compiled into an R package, and we're distributing it around the country. so that other hospitals and other blood centers can use the software on their data and make predictions in their blood center. And it's in place now at Stanford Blood Center, and it's being monitored now. It's not actually being used to make actual calls, but it's being monitored to see how well is it doing in predicting how much blood is going to be needed. Right? We want to be careful about this, because if you put a system in place and it starts to screw up, then people sort of they lose patience with algorithms, right? And another example of this is like self-driving cars. Now you see in the newspaper, a Google self-driving car had an accident after like 2 billion miles or something of testing, right? Humans have accidents far more often, but somehow it's okay for humans to have accidents, but self-driving cars, even if the error rate, the accident rate is far, far lower, we're not very forgiving when machines make a mistake. Same here, right? There's been a lot of waste of blood, but it's been done by humans, that's okay. But we want to make sure that the machine algorithm, the statistical algorithm, does a good job, because otherwise people will not want to adapt it. People don't trust sort of statistical machine learning algorithms. They tend to have a very low tolerance for their mistakes, basically. So, questions about this or comments? So this is an example of sort of statistics in action, right? You get some data, you fit a model to it, and use the model to help guide decisions.
I was wondering, so do you continue monitoring it as well to see if you're able to reduce it even further, or do you just accept it at that level and say that level is okay?
Well, we do monitor it. So now since you've asked that question, I'm going to come clean and say, I think maybe you're the same person who asked the question earlier. So we have it, I said this already, basically, we're distributing the software, but recently, it's actually not this week, it's now this month. So we have the system in place, but we're not using it for actual decisions yet. We're just sort of running it in parallel with the human ordering system. But we noticed at one point in the last few months, actually this is sort of before COVID started, because we haven't been able to get back to it, it started really screwing up. Its predictions were way off. And then someone asked the question at the beginning, what happens if your data is wrong? I suspect what happened is that the incoming data to the system from the hospital database has got some kind of a corruption. It's corrupted, or someone's changed the format or something, because the model was making very bad predictions. So soon we're going to get back to this and monitor it. But this is actually a big general question for statistical systems. How do we detect when a system's no longer working well? So your question was, do we monitor to try to improve it? Well, even more important, do we monitor to make sure it's not messing up, right? Like you build a system that works well with historical data, you put it in place, you start to use it, and if it starts to mess up, you have to have a way of detecting that automatically. Tell the user, hey, wait a minute, your system is making bad predictions, right? So this is really an important part of whenever you try to use a statistical system for doing predictions, and this is happening in a lot of different areas, you have to have a way of monitoring its quality. Right? And I mean, this is, I know someone who works for Bombardier, and they write software for detecting, when a train's going around the track quickly, a fast train, right? When when the speed is, they make sure the speed stays in a certain range so the train doesn't derail. Well, it's quite remarkable. They actually have two software systems for doing this detection. And the software systems are written in different languages, on different hardware, on different chips. And they both have exactly the same job, but they run in parallel. And as soon as everything's great until the software systems all of a sudden don't agree, then they sound an alarm. Again, because there's a lot at stake if a train derails, right? But they go to it's possible that there's a problem, maybe the chip has a flaw. So real-time systems have a lot of safety checks built in to make sure whatever the job is, detecting anomalies, detecting, a train going too fast, they have to work and they have to be monitored for quality. So, okay, let me ask you guys a question. So we have this prediction equation now, right? Which does this prediction and it's working well at Stanford. Can I just ship this prediction equation across to Massachusetts, and can they use it as is in Massachusetts or Texas, or what do you think? I said we're sharing software. Should we just share the prediction equation, or do we need to do something different? So we have a certain prediction equation that works at Stanford. Can I assume it's going to work in every state in the country? That same equation?
No, I think we should like check on their local conditions and other factors to see how does that affect the result.
Right, good, very good. In statistical terms, we're going to retrain the model. So we're going to, this process, we ship the algorithm to Massachusetts or Texas. They have to run, they take their own training data, their own patient data, right? Because their patients won't be the same as ours. They'll have, you know, maybe it's a big city hospital, a lot of, I don't know, younger or older people, just people are different than we are. So they'll take the algorithm, but they'll retrain it on their own data and make their own predictions. So that's an important point. You can't just use prediction equations across different states or cities. The model and the algorithm are valid, but they have to be trained on each of the individual, you know, in your hospital. So the idea is we're going to ship the algorithm in our package with instructions. You have to collect the following data, train the algorithm on your data to make your predictions. Okay, so yes, that's an important point, that prediction equations are not just, you can't transfer them from one to the other. So I think I had a second section about COVID forecasting, but I'm not going to have time to go over that because I think we're out of time. But if you want to read about it in the slides when you get them, you're welcome to. But what it's about, and that the pointer is in the notes you're going to get. I've been involved in this in a COVID map. So this is Carnegie Mellon University, where my son is a professor, and he started this. I'm part of a team that produces this COVID indicator map. And it's been a fun, important project and a lot of work. And it displays things like symptoms of COVID, cases of COVID, doctor's visits, and you can look by state and by city, etc. So this is called COVIDcast. And there's a lot of statistical forecasting involved in this project. So sorry I didn't get time to talk about this, but a lot of the tools I talked about today, this prediction equation ideas, is inherent in these kinds of maps too. They're trying to predict how many cases, say, is Santa Clara County going to have of COVID next week. Well, we forecast that, for example, how many cases did it have this week? How many people are reporting symptoms? And other factors. So again, it's a supervised learning problem. Build a prediction equation from historical data to try to forecast the future. Okay, so that's called COVIDcast, and the details of this are in the notes, which I'm given to Ismael. So I will just cut to the very end. I hope at the least you got some flavor of what modern statistics is about. And if you want to take more statistics classes or read more, these are books which some of these first we have co-written. They're all free, so I don't feel bad about advertising. They're all free online. And these sort of go into more and more details. This first one especially might be interesting to you if you want sort of a next course in statistics and modern statistics. This book, Introduction to Statistical Learning, which is an online PDF form, might be of interest to you. And there's also a MOOC Stanford course that two of us, Professor Hasty and I, give on this topic. So it's sort of the next level course after Stat 60, or maybe a couple courses later. But if you're interested in learning more about modern statistics in more detail, then this would be a good book to look at. Okay.
Thank you very much for this.
If you want to go, that's fine. If you have questions, I can hang around too.
I actually had also a few questions. So you mentioned some of the classes that students could take? What are some of the other ways that students can prepare themselves for, for this data revolution? Are there any opportunities for students to get involved, say, in this COVID cast or other projects? And just how should one approach this besides classes?
Well, I think getting involved in a project is always good, yeah. The COVID cast, we don't really have, there are other opportunities on campus. There's lots of projects, COVID projects on campus, and they're often looking for help from students. But the one thing is you need programming skills. So if you guys have learned R, so I guess one thing is I would continue to upgrade your R skills and/or learn some Python, because that's really what will make you useful to people. There's lots of opportunities on campus for these kinds of projects. So, any other questions? Anybody?
In university, I want to study statistics. In university, will we relearn how to use R, or should we be already sophisticated with it?
Oh, well, I taught Data Science 101 last year, and we started teaching R from scratch. So it depends on the course. If you take this course here, for example, this is Stat 215. We kind of expect you to know R, or at least learn it in a week or something. So it certainly doesn't hurt to come into the class with some good background in R. And there are lots of online courses that can, for example, again, we have an online course with exactly this title, Trevor Hasty and I, on MOOC, in which it's free. There's a YouTube version of it, and you can just go through the course, and along the way you learn to do R and apply R in lots of different settings. So I find it's easiest to learn by doing. Like, it's hard to learn a language in the abstract. It's much easier if you actually have to you're doing something, right? You're asked to solve a problem. So, yeah.
I think we also have quite a few students from a social science background. Is there anything specific that these students should know as they take these classes and they want to apply them to their own field of research, which might not be statistics?
Well,
psychology and other fields.
I can tell you that statistics is becoming, a lot of these, like psychology, sociology, becoming very quantitative. Everybody's collecting data in every field now. So what used to be the soft sciences are really becoming much more data driven. So even if you think you're not doing hard science, you find data science is more and more important because everybody's collecting data. It's easy to do, and you can get information. So sociology, psychology, other fields like that, they're all much more data driven than ever before.
Let's thank once again Professor Chipshirani for the guest lecture today. I have made the slides available on the course website, and they include all of the other material that Professor Chipshirani mentioned. I think that's all I had to say for today. This is the end of the lecture, and I will be holding office hours right away on the other Zoom link. And I will see you there if there's anything, if you have any concerns that you'd like to discuss.
Okay.
Thank you all.
Okay.
Thank you.
Thank you.
Oh, thank you.
Thank you.
Thanks for listening.